Designed computationally and refined in the laboratory, the new protein therapies thwarted infection by interfering with the virus' ability to enter cells. The top protein neutralized the virus with similar or greater potency than antibody treatments with emergency use authorization status from the U.S. Food and Drug Administration, FDA. The findings were published yesterday, April 12, in the journal Science Translational Medicine. This work was led by Northwestern's Michael Jewett, David Baker and David Wiesler at the University of Washington School of Medicine, and Michael S. Diamond at WashU. To begin, the team first used supercomputers to design proteins that could stick to vulnerable sites on the surface of the novel coronavirus, targeting the spike protein. This work was originally reported in 2020 in the journal Science. In the new work, the team re-engineered the proteins, called minibinders, to make them even more potent. Rather than targeting just one site of the virus infectious machinery, the minibinders simultaneously bind to 3 sites, making the drug less likely to detach. The interaction between the spike protein and our antiviral is among the tightest interactions known in biology. Andrew C. Hunt, a graduate research fellow in Jewett's laboratory, is the paper's co-first author. As the SARS-CoV-2 virus has mutated to create new variants, some treatments have become less effective in fighting the ever-evolving virus. Just last month, the FDA paused several monoclonal antibody treatments, for example, due to their failure against the BA.2 Omicron subvariant. By blocking the virus spike protein, the new antiviral prevents it from binding to the human angiotensin-converting enzyme 2, ACE2, receptor, which is the entry point for infecting the body. Because the novel coronavirus and its mutant variants cannot infect the body without binding to the ACE2 receptor, the antiviral also should work against future variants. To enter the body, the spike protein and ACE2 receptor engage in a handshake, Jewett said. As opposed to monoclonal antibodies, which are made by cloning and culturing living mammalian cells, the new antiviral treatment is produced large-scale in microorganisms like E. coli, making them more cost-effective to manufacture. Not only is the new therapy stable in high heat, which could further streamline manufacturing and decrease the cost of goods for clinical development, it also holds promise for being self-administered as a one-time nasal spray, bypassing the need for medical professionals. This study, multivalent designed proteins neutralize SARS-CoV-2 variants of concern and confer protection against infection in mice, was supported by the Audacious Project at the Institute for Protein Design, Bill Melinda Gates Foundation, OPP 1156262, INV004949, Burroughs Wellcome Fund, Camille. Make a Difference. Sponsored Opportunity StorySource. Materials provided by Northwestern University. Original written by Amanda Morris. Note: content may be edited for style and length. Green, Wadham L. Matochko, Christy A. Thompson, Bastian Vogeli, Ant J. Kruger, Laura A. Van Blargen, Rita E. Chen, Bailing Ying, Adam L. Bailey, Natasha M. Kafai, Scott E. Boykin, Ajasha Lubetic, Natasha Edman, George Ueda, Cameron M. Chow, Max Johnson, Amin Adisha, Mary Jane Navarro, Nutada Panprati. New COVID-19 nasal spray outperforms current antibody treatments in mice. A single inhaled dose treated or even prevented infection by COVID-19 and its variants. Science Daily. New COVID-19 nasal spray outperforms current antibody treatments in mice. A single inhaled dose treated or even prevented infection by COVID-19 and its variants. Science Daily. https://ift. TT, SZLJDUT, accessed April 13, 2022. Thanks for watching. Please subscribe my channel for more videos.